Imagine a world where medicines don't just treat symptoms, but target the exact molecule causing the disease. A world where drugs are designed, not discovered. Where cells become factories. Where antibodies become weapons. Where biology becomes programmable. This is the story of how scientists learned not just to read DNA, but to engineer life itself. Welcome back to ScientificCareer. This is episode 7 of our 10-episode journey through the history of the pharmaceutical and biotechnology industries. Today, we explore the rise of modern biotech, monoclonal antibodies, engineered proteins, targeted therapies that reshaped medicine. Let's begin. By 1980s, scientists understood DNA. They knew how genes worked. They knew how to cut and paste genetic material. They knew how to engineer bacteria to produce human proteins. But they wanted more. They wanted medicines that could target disease with precision. They wanted therapies that could recognize cancer cells. They wanted drugs that could block specific molecules inside the body. The next revolution wasn't about DNA. It was about proteins. In 1975, two scientists, Georges Kohler and César Milstein, made a discovery that would change medicine forever. They created the first monoclonal antibody, a molecule that could recognize one specific target, one protein, one receptor, one antigen. It was like giving medicine a guided missile. But there was a problem. These early antibodies came from mice. The human immune system rejected them. They caused reactions. They didn't last long. The idea was brilliant. The technology wasn't ready. Not yet. Throughout the 1980s and 1990s, scientists developed new ways to humanize antibodies. They replaced mouse components with human DNA. They engineered antibodies that the immune system would accept. They created molecules that could stay in the bloodstream for weeks. This breakthrough opened the door to a new class of medicines: cancer immunotherapies, autoimmune disease treatments, anti-inflammatory biologics, targeted therapies for chronic illness. Biotech companies saw the future and raced to build it. One company would lead the way. Amgen, a young biotech startup in California, set out to engineer a protein that could help patients with kidney failure. These patients suffered from severe anemia. Their kidneys couldn't produce erythropoietin, the hormone that tells bone marrow to make red blood cells. Amgen used recombinant DNA technology to engineer EPO, a synthetic version of the natural hormone. In 1989, the FDA approved Epogen. It became one of the first blockbuster biologics in history. Epogen transformed dialysis care. It generated billions in revenue. It proved that biotech wasn't just a scientific revolution, it was a business revolution. And it showed pharmaceutical companies that engineered proteins could outperform traditional drugs. But the most dramatic breakthrough came from a company called Genentech. In the 1990s, scientists discovered that some breast cancers were driven by a protein called HER2. Tumors with too much HER2 grew faster, spread faster, and were harder to treat. Genentech developed a monoclonal antibody that could target HER2 directly. They called it Herceptin. Herceptin didn't just slow cancer. It changed the survival curve. It gave patients years of life they never would have had. It was one of the first true targeted cancer therapies, and it proved that monoclonal antibodies could do what chemotherapy never could: attack cancer cells while sparing healthy ones. By the 2000s, monoclonal antibodies and engineered proteins were everywhere. They treated rheumatoid arthritis, Crohn's disease, psoriasis, multiple sclerosis, lymphomas, leukemias, breast cancer, colon cancer. Medicines like Rituxan, Remicade, Avastin, Enbrel, Humira, Keytruda. These weren't pills. They were biologics. large, complex molecules produced in living cells. They didn't just treat disease, they targeted the exact pathways causing it. This was precision medicine before the term even existed. The rise of biologics reshaped everything. Pharmaceutical companies built massive bioreactors. They invested in cell line engineering. They created new regulatory pathways. They developed cold chain logistics. They built entire divisions around monoclonal antibodies. Biotech wasn't a niche anymore. It was the future. By the 2010s, biologics accounted for many of the world's top-selling drugs, and the industry was preparing for the next leap: gene therapy, cell therapy, genome editing. But that story belongs to Episode 8. The rise of modern biotech teaches us that the biggest breakthroughs happen when science and engineering collide. Antibodies, recombinant proteins, engineered cells. These technologies didn't replace traditional pharmaceuticals. They expanded what medicine could do. They turned diseases once considered untreatable into manageable conditions. They transformed cancer care. They created new careers, new industries, and new possibilities. And they set the stage for the next revolution: editing the code of life itself. If you want to keep learning about the incredible world of medicine and build the skills to thrive in one of the fastest growing industries on the planet, make sure you like this video, leave a comment, and subscribe to ScientificCareer. Your journey in science and healthcare is just beginning, and we're here to guide you every step of the way. But engineering proteins was only the beginning. Because once scientists learned how to design antibodies, they wanted to design cells. They wanted to rewrite genes. They wanted to cure diseases at the source. In the next episode, we explore the rise of gene therapy, cell therapy, and CRISPR. Technologies that give us the power to edit the code of life. Episode 8: The Gene Editing Revolution. Don't miss it.